Good day. Today I will be discussing the metabolic responses of Caenorhabditis elegans following exposure to two metal-based nanomaterials and their bulk counterparts. Metal-based nanomaterials are increasingly implemented in many products. Consequently, they may ultimately be released into the environment during production, use, and disposal at the end of their life. Quantum dots are of great interest due to their tunable optical properties, which are used for multi-target imaging. A typical quantum dot is composed of a semiconductor core, in this case cadmium beryllium, that can be encapsulated with functional side chains, in this case ammonium. Nanoscale copper oxide was also chosen as a case study because it is widely used for antibacterial properties in wood preservation, paints, and textiles, and copper is also a known toxicant in the aquatic environment. Metabolomics has been applied to both the terrestrial and aquatic environments to investigate the effects of toxicants, disease, and other environmental stresses on organism health and metabolism. Specifically, non-targeted metabolic analysis of samples where the goal is typically to discover biomarkers that discriminate between different groups of samples, for example, the control versus stressed. Metabolomics can provide possible mechanistic insight into nanotoxicity. To this date, there are only two studies using C. elegans and nanomaterials looking at metabolomic responses: titanium dioxide and polystyrene. C. elegans is abundant in soil ecosystems and also found in aquatic and sediment environments. It is easy to culture, has a short lifespan, and has a well-characterized genome. So the aim of this study was to establish the metabolic response of C. elegans after exposure to metal-based nanomaterials and their bulk counterparts. The exposure was carried out by bleaching 120 agar plates seeded with nematodes. After eggs hatched, L1 stage nematodes were then exposed to the nanomaterials and their bulk counterparts individually at concentrations equal to that of their EC50 for reproduction. After exposure, nematodes were collected via centrifuging suspensions and discarding the supernatant. These suspensions were then put through a sucrose separation step to separate, settling out nanomaterials from the nematodes. Extraction was done using a standard method by Beerkus 2019, and samples were read on a GC-GC time-of-flight mass spectrometer. Quality control samples were applied to demonstrate analytical accuracy, precision, and repeatability. Relative concentrations were corrected according to initial sample weight, and the relative concentrations are therefore expressed as nanograms per milligram sample. Data was analyzed using MetaboAnalyst, and normalization and auto-scaling was performed. ANOVA and t-tests were performed to test for significant metabolites. From the one-way ANOVA results, a PLSDA2D scores plot was generated for nanomaterials versus the control, as well as bulk materials versus the control. From here we can see a clear grouping and separation between the nanomaterials and the control, the green being the control on both graphs. In the bulk material graph, there is slight overlap between the control and cadmium in red and beryllium in light blue. To further establish the reason for these groupings, pathway analysis was performed. As a rule, only two groups can be compared using this function in MetaboAnalyst. Therefore, each material was compared to the control. First, copper oxide was analyzed. Compared to the control, there were 24 significant metabolites. However, only 15 of these could be identified. On the right is a representation of some of these metabolite concentrations. As you can see, the alpha-linolenic acid concentration was much lower in copper oxide than in the control. Alpha-linolenic acid is part of the alpha-linolenic acid metabolism, which is part of the lipid metabolism and protects lipids from reactive oxygen species. The lipid metabolism regulates fat content and increases longevity or lifespan. Thus, lower concentrations of this metabolite could eventually lead to lower life expectancy. D-xylose forms part of the pentose and glucuronate interconversions pathway, part of the carbohydrate metabolism. This metabolism is responsible for metabolite detoxification and excretion and leads into the citrate cycle, responsible for ATP production. Bulk copper revealed 45 significant metabolites where only 7 could be identified. Sucrose was a large influence on the separation observed on the PLSDA on the left. Sucrose was much lower in the copper-exposed group than the control. Sucrose forms part of the starch and sucrose metabolism, responsible for lifespan. This metabolism also links to the amino sugar and nucleotide sugar metabolism, and glycolysis, responsible for the biosynthesis of carbohydrates and conjugates. This is in larger part of the carbohydrate metabolism. Sucrose also forms part of the galactose metabolism, responsible for D-fructose production, which leads to energy production. Quantum dot and its bulk materials, cadmium and beryllium, did not have enough significant metabolites to do formal pathway analysis. I will, however, be reporting the significant metabolites and the pathways they form part of. In the case of quantum dots, phenylacetic acid, eicosapentaenoic acid, propylene glycol, pyranton, and oleic acid were considered significant, all lower than the control. Phenylacetic acid is part of the phenylalanin metabolism, which makes tyrosine, an essential component of neurotransmitters. It also forms part of the xenobiotic biodegradation pathway. Eicosapentaenoic acid is part of the biosynthesis of unsaturated fatty acids and part of the lipid metabolism. Polyunsaturated fatty acids are precursors for signaling molecules. Products from these metabolisms have severe biological effects, such as pharyngeal pumping, growth, and reproduction effects. Polypropylene glycol is part of the propanoate metabolism that leads into the citrate cycle and forms part of the carbohydrate metabolism. Oleic acid also forms part of the fatty acid metabolism, biosynthesis of unsaturated fatty acids, as well as the longevity regulating pathway shown in the image. It can cause the organism to essentially shut down reproductive outputs in order to expand their lifespan. The image shows germline removal with a red arrow, results in increased lifespan and stress resistance. This makes sense due to the fact that the concentration used was at the EC50 of reproduction. When looking at cadmium, 3 identifiable out of 7 total metabolites were responsible for the grouping seen on the left: trimethylamine, 4-T-butylbenzoic acid, and D-glucose. They were all significantly higher than the control. Trimethylamine is part of the carbon metabolism and acts as a nutrient signal for the formation of dire larvae, modulates food reception, and influences lifespan. As controls were exposed under the same conditions without food, this reaction is likely due to another toxicity pathway increasing dire larvae formation time compared to the control, which also influences reproduction. D-glucose is part of the starch and sucrose metabolism, glycolysis, and galactose metabolism that forms part of the amino acid and sugar nucleotide metabolism, which affects starch and sucrose metabolism, which is responsible for energy. Tellurium revealed 4 identifiable metabolites out of 7 total: D-xylopyranose, D-fructose, 2-tridacol, propylene glycol. All 4 were significantly less than the control and completely inhibited. D-xylose and D-fructose form part of the amino sugar and nucleotide sugar metabolism. D-xylose was also seen to be inhibited in the copper oxide group. Propylene glycol forms part of the propanoate metabolism and was also inhibited in the quantum dot group. The only overlapping metabolites were D-xylose between beryllium and copper oxide, as well as propylene glycol between beryllium and quantum dots. Non-overlapping metabolites can thus be possible biomarkers for nanospecific reactions. Cadmium was also the only material that caused a significant response in the xenobiotic pathway. Common pathways that were affected are lipid and carbohydrate metabolism pathways. These two pathways affect nearly all aspects of organisms' biology, and disruption could be devastating. They also link with each other. Energy production and longevity were seen as common outcomes of these pathways. No pathway top-down effects were observed, but exposures were only 24 hours long, and longer exposures could lead to more severe top-down pathway effects. These results, however, could still indicate early response biomarkers for C. elegans exposure. These early biomarkers could include biomarkers from the fatty acid and lipid metabolism for nanomaterials, such as alpha-linolenic acid and oleic acid. For bulk materials, it could be carbohydrate and starch and sucrose metabolism with metabolites such as D-glucose and sucrose for bulk materials. Thank you.